Hello, this is the AP Biology Enzyme Lab that we are going to run today. I have everything set up here, so I'm going to start mixing the substrate and the enzyme in just a minute. But before we start, some things you should have looked at already. Possibly the video with the background lab that had the celery and the hydrogen peroxide. Definitely you should have read through the background that was shared through AP Classroom or Google Classroom about the guaiacol and the hydrogen peroxide and the role of the peroxidase from the turnip. Hopefully also you have a data table ready to go that is going to show the color change of our reaction over 5 minutes. So we can talk about that when we're a little bit closer to time. But let's go ahead and get started. Just to go through a few of the things that I have out here, I have some distilled water. So we're going to use those, of course, in just a minute. I have one of my tubes that I've labeled with an S, and that's for the substrate. And the substrate in this lab is the hydrogen peroxide, which I have right here. In the brown bottle, probably looks like a little tiny version of the hydrogen peroxide you have at home in your medicine cabinet. Same thing, this is just less concentrated, but it's still hydrogen peroxide, the H2O2. When you break this down, this will break down into water and oxygen gas. It will do that on its own over time. We've talked about this a little bit in the background. That's why most hydrogen peroxide bottles have expiration dates on them. However, if you add an enzyme, that reaction will be catalyzed and happen more quickly. So that's what we're going to do today. So we'll put the hydrogen peroxide in our substrate tube, and then also in that tube, in addition to the water to bring it up to a certain volume, we're going to add the guaiacol. The guaiacol in this reaction is only the indicator. Whenever oxygen is produced, it combines with the guaiacol to produce a brown precipitate color. So as we're watching the color change, what that's actually showing us is how much oxygen is being produced. And we can determine an enzyme rate from that. So in the substrate tube, we'll have the substrate, which is the hydrogen peroxide, the water to bring it up to a common volume, and then some of the indicator. Okay, so I'm going to go ahead and put that in there. Hopefully you read through the procedure that was shared with you, or you have it available in front of you right now. So you know we've got our water in there, and then I have my 2 plastic pipettes, and I've labeled them so I won't get them confused. So I'm going to add the hydrogen peroxide. Both tubes should come up to a volume of 7.5 milliliters. So you want that to be a constant in our experiment. So there's our hydrogen peroxide. Okay. And then we're going to add the guaiacol using another dropper. There's really no safety concerns in this lab, except the guaiacol can be an irritant if you get it on your skin or in your eyes. But I am wearing my glasses, so I'm not worried about that, and I'll put on my gloves when I do the mixing because I don't have any parafilm for the top. So that's everything that's going to go in our substrate tube. Now let's talk about the enzyme tube. Only 2 things are going in here. We are adding some distilled water. Again, we want, I spilled a little bit there. We want them to be approximately the same volume. And then in here I have the enzyme. So this was made by taking a turnip and creating essentially a slurry in a blender. Plants produce a lot of peroxidase because hydrogen peroxide is a waste product in plant cells and they need to get rid of it pretty quickly. So this is essentially turnip juice, and we are going to use it to supply our enzyme. So again, I'm going to add that to this test tube, and they should be approximately the same volume. I'm going to put on my gloves. Again, the concentration of the glycol is pretty weak, but just to be safe, ideally you would have a little cork or a little piece of parafilm so you could mix the tubes. I'm just going to use my gloves like this, give them each a little shake. Now, before the next step, things are going to start happening pretty quick. So you want to make sure that you have, if you want to pause, you want to have your data table set up. If you've read through the procedure, you've seen that we are going to time this for 5 minutes, and every 30 seconds to a minute, you can decide how you want to set up your data. You are going to record the color of this solution. So I'm going to mix the substrate tube and the enzyme tube, and you think about what will happen to this hydrogen peroxide substrate when the enzyme mixes with it. And we are going to use this color scale. So this was shared with you in the lab. I'm gonna leave it up here. I'm gonna try to set this up so you can collect your own data with the timer and the color scale. So at each, I would say 30-second interval for 5 minutes, you are going to record the time and the number on this scale that corresponds with the test tube. Now pause for a moment and think about what that means. As the test tube is getting darker, as there's more of that brown color being produced, what does that mean? Okay. All right, so give me just a minute while you're working on your data table and thinking through some of those things. And I'm gonna move the camera and get a little bit close up so you can see the color and the timer. Maybe. I'm also going to put a white piece of paper behind this for our reaction because I think that makes it easier to see. Okay, let me move our camera. There we go. So you'll have the test tube directly in the middle, and then you're going to have the color chart to the right and the timer to the left so you can collect your data. Okay. Again, I'm going to pour them together and give them a little mix, and then I'll start the timer. Okay, here we go. Okay. So again, we're at about 12 seconds. At 30 seconds, you would record a number on the color scale. "30 seconds. I need some music playing like in the Bozeman videos. My production values are a little lower. Coming up on a minute. Zoom in if you need to, pause it. Refer to your own color chart that was shared on the computer. Okay, coming up on a minute 30. Hopefully you can see the timer and the test tube and the color chart. Coming up on 2 minutes. Again, the procedure from College Board says every minute, and that's fine. I think the graph looks a little bit nicer if you have every 30 seconds. So all that's in this test tube is the hydrogen peroxide, which remember is clear, the distilled water, the guaiacol, also clear. Everything was clear in those tubes until I mixed them. Coming up on 3 minutes, if you'll judge your color there. You may get to a point where you think it's kind of stabilized and it's not getting any darker. That's fine. Let's keep recording data points for the 5 minutes. From my angle, I can't see it very well, but hopefully you can judge the color on the scale. Since we're coming up on 4 minutes, we're probably approaching the far end of the brown color coding. And 1 more minute. Again, some things you want to be thinking about. Why do we stop the timer after 5 minutes? Why does the color scale only go to 10? If I were to let this sit overnight and come back tomorrow morning, would it be super dark? A lot of those things, if you go back and look at some of the controls on enzymes, there we are at 4.5 minutes. As far as concentration of substrate, product buildup, some of those things, you'll understand why we only go for 5 minutes and we don't need a color scale much darker than 10. All right, and we're coming up on our 5 minutes. So for your final data point, go ahead now and record what you think that final color would be. And that's the end of the enzyme lab. If you would refer to the instructions online for the write-up, and you are going to create a graphic of this data, so think about what would go on your x-axis, what would go on your y-axis, and then we're also going to run a variable. So same procedure, but change something. So I'll have that data shared in the next video.